We stopped at the relationship of the DNA-based sequence to the peptide structure. So we have a start codon, AUG, and we have a stop codon, U go away, UGA, U are away, UAA, and U are gone, UAG. That's the stop codon. So what's the name of this structure here? What's the name of this RNA that is carrying for us these peptides, this amino acid? What is that called? What is this hairpin-like structure called? The tRNA, the transfer RNA, transferring for us the amino acids. Okay, so you have to remember that there are ribosomes at the surface of the rough endoplasmic reticulum, and there are ribosomes free floating in the cytoplasm. What's the difference? What's the difference? Why is there a class of ribosomes embedded on the surface of the rough endoplasmic reticulum and ribosomes free floating in the cytoplasm? Both of them make proteins, but the proteins that are made on the rough endoplasmic reticulum are going to be duct, you see, to the inside of the rough endoplasmic reticulum, and then a vesicle is going to exocytose, and it's going to be received by the Golgi body, and then the Golgi body is going to package it. It's going to receive it through the cis side and package it and send it out through the trans side to the outside of the cell. So rough endoplasmic reticulum ribosomes make proteins to go outside of the cell. Rough endoplasmic reticulum proteins make rough endoplasmic reticulum bound ribosomes make proteins that are to be received by the Golgi body at the cis side, and it will be exported through the trans side of the Golgi body to go outside of the cell, to be used outside of the cell. They're not for local usage. Okay, post-translational modification is when we make our tRNA. That is when we make our ribosomal proteinaceous protein, and this protein now needs to be modified before it is sent out. It needs to be modified. And we can activate, so for example, if we examine here, we have this mechanism of gene activation where prolactin hormone, a polypeptide hormone, is going to bind to the prolactin receptor. Then the prolactin receptor is going to dephosphorylate ATP and bind one phosphate group to a regulatory protein, transcription factor. That transcription factor activator is going to go inside the cell to the DNA, and that's going to transcribe for us. You know, the casein gene is going to be activated, and then RNA polymerase is going to transcribe the casein gene. Then the casein gene is going to be translated by the ribosomes that are on the rough endoplasmic reticulum, which will be sent out into the outside of the cell. This is an example of how rough endoplasmic reticulum bound ribosomes make proteins to be used outside of the cell, like casein. Casein is part of the milk in the milk duct to make the milk for the baby. Now, you also have indirect control of testosterone synthesis by the DNA. So number one, DNA is going to code for the mRNA, and then the mRNA is going to code for the synthesis of some enzyme. This enzyme is going to be inactive. But when testosterone is needed, a very important hormone known as luteinizing hormone, LH, is going to stimulate the production of a second messenger within the cell. And this second messenger is going to activate enzymes. And these enzymes are going to convert for us cholesterol into testosterone. That's why we said there are cholesterol-based hormones like testosterone, progesterone, aldosterone, estrogen, and then we're going to have testosterone in the end. Okay, so for replication, we discussed replication. Replication is a semi semi-conservative and a discontinuous process. Semi-conservative and a discontinuous process. Why is that? Because you have your DNA double helix, so you need this orange enzyme here. What do we call that? That's DNA helicase. Helicase is going to unwind for us the DNA double strand. That's its job, like a unzipper. It's going to unzip for us the two strands that are bound to each other. Then you have two strands. What's going to happen now is you see this big green enzyme here. What is that? This is the DNA polymerase. It's going to be making DNA for us. And you know the DNA is anti-parallel. So the DNA is in fact made up of two strands that are bound to each other. We talked about this earlier with the hydrogen bonds between the two strands. And we said that they are anti-parallel from 5' to 3', and the other side is from 3' to 5'. So if your polymerase is transcribing in one direction from 5' to 3', the 5' to 3' on the other direction has to be on the opposite direction. This is why we will have gaps in the replication. So these gaps are known as Okazaki fragments, named after the Japanese scientists who discovered them. These Okazaki fragments are going to be bound together by an enzyme known as DNA ligase. Ligate, ligation, glue, gluing together, ligating those empty fragments. So do you see DNA ligase? So what is the job of DNA ligase? It's going to glue together the Okazaki fragment. Now, why do we call the DNA discontinuous? Because you have Okazaki fragments. On one side, you don't have fragments because the direction of transcription is 3' to 5'. But here, the direction of transcription is 3' to 5' because the DNA is anti-parallel. So it will be discontinuous. There will be gaps that must be replicated later on. So that's why we call it discontinuous. Why do we call the DNA replication semi-conservative? We call it semi-conservative because when helicase unzipped for us the DNA, it unzipped it into two different strands. But from each strand, you made a copy. So you had two parental strands, and from each parental strand, you made a daughter strand. That's why we call it semi-conservative, because you don't have two parental strands. You have one parental strand and a copy of that, the daughter strand. And here you have a parental strand and a copy. You see, you have the daughter DNA and the old strand in both, the daughter DNA and the old strand. That's why we call it a process that is semi-conservative and discontinuous. When the DNA is damaged, we talked about when proteins are damaged, you have the proteasome that's going to degrade all the misfolded, improperly folded proteins. Also, when the DNA is damaged, we need to replenish it and remove the damaged nucleotides and replace them with better, more healthy nucleotides. Mutations are changes in the DNA structure. It could be because of metabolic stress. It could be because of ultraviolet radiation. That's why sunlight causes cancer, because it damages the DNA. So you have this melanin. This dark color is like a sunscreen to protect the DNA from the damage. So the cell cycle is very important to know. I ask my students, what are the two phases of the cell cycle? And they say prophase, metaphase. No, those are the phases of mitosis. So the cell cycle has two phases. The brown part, the small part, mitosis, and the very big green part known as interphase. That's very important to know. The cell cycle is made up of mitosis and interphase. Now, each one is divided into separate phases. How many phases is interphase broken down into? 3 phases. You need to know those. The G1 phase, that's when the cells grow and they adjust their normal metabolic rate. And the S phase is when the DNA is replicated. You have doubling the DNA to prepare for division. And lastly, you have the screening of this DNA that we did not double up a DNA that is damaged. We don't want to double a DNA that is mutated. So we screen the DNA, we grow, we prepare for mitosis. Then we enter the mitotic phase. You will see that the same way, each phase of interphase had the distinctive feature. So you need to know what are the distinctive features of G1, S, and G2. G1's distinctive feature is that it's the phase of the growth and the normal metabolic functions. The S phase is the phase where there is the DNA replication. And lastly, you have the second gap phase, and that is the growth and the preparation for mitosis. Then once you enter mitosis, you have prophase, metaphase, anaphase, telophase, and cytokinesis. You need to know the distinctive features of each of these phases in mitosis. Prophase is known for what? Is known for the fragmentation and the breakage of the nuclear envelope, the nuclear membranes, known as karyorrhexis. Karyorrhexis means we destroy the nuclear membrane. In fact, we need to do that because how are we going to divide the DNA and the nuclear membrane is still there? We need to remove the nuclear DNA. Then in metaphase, we line up the chromosomes at something known as the metaphase plate. What's the metaphase plate? The metaphase plate is an arrangement where we line up all of the X's, the chromosomes, at the middle of the cell. Then anaphase is the most easiest one to identify because you see that the plucking off. You see these two sister chromatids came apart because the microtubules attach to the kinetochores and they are pulling in opposite directions. Telophase is the appearance of the cleavage furrow and the breakage of the two cells, and then cytokinesis is the actual division of both cells. Is that clear? Of course. Of course. You know, you think we are just doing like a storytelling? Of course, you need to know all of that. Okay. Mitotic is not that difficult though. If you just go over it, it's very, very counterintuitive. It's very counterintuitive. It's very straightforward and it's not counterintuitive. Okay? G0 phase. Those are the cells that do not. G0 phase. Cells that what? I talked about them earlier. What were they? Cells that don't divide no more. Excellent. The nervous tissue and the cardiac tissue. So if we look here at prophase, look at prophase. What happens? The nuclear membrane, the nuclear envelope breaks down. That's the main feature of that prophase phase. Metaphase. What do you have? What plate do you have? In metaphase, what do you have? What do we call this plate here? It's called the metaphase plate, where the chromosomes lie along the midline of the cell. Anaphase is when they both separate. Telophase is when the cleavage furrow starts to appear and the daughter cells eventually divide. The karyotype is our chromosomes. 1 to 22 represents the autosomal cells, and 23 tells you these are the sex chromosomes to tell you if you are X and X means you're a female, X and Y it means you are a male. So all the cells are diploid. They have 46 chromosomes, except the sex cells, the gametes. Sperms have 23, and the egg has 23. They join together to give you 46. Okay, so genes and alleles. There are dominant alleles and recessive alleles. This is very important for sickle cell anemia. If you examine sickle cell disease, is a disease that is caused by an abrupt defect, structural defect of the red blood cell structure. So the red blood cell must be discoid like a disc. The red blood cell, in addition to being discoid, it must also be what? Biconcave. biconcave, depressed from both sides, and discoid and biconcave. There are proteins, cytoskeletal proteins like spectrin, that ensure that your red blood cells are discoid and biconcave. People who have sickle cell disease, they are born with a hereditary defect where something is wrong with these proteins, cytoskeletal structural proteins. So the structure of the red blood cell is no longer biconcave, is no longer discoid. It looks like a sickle, sharp structure. So how do you get that disease? You have to be homozygous recessive. If you are a homozygous dominant, two capital H's, then you will be normal. If you have a dominant allele and a recessive allele combined together, that's heterozygous, but you're a carrier. You don't have the disease, but you're a carrier. But if you end up with a child who takes, let's say you're a carrier and your husband is a carrier, then the child gets a sperm, is made up due to a sperm that is allele, a recessive allele, and a recessive allele from the egg, and then you have two recessive alleles, then the baby will not have biconcave discoid structure red blood cell. The baby is going to unfortunately have sickle cell anemia. By the way, if you have two people, one with sickle cell anemia while the other one is healthy, and both are relaxed and sitting down, taking deep breath, enjoying, watching the television, you take a blood sample from both of them. Will you be able to tell which one has sickle cell anemia? If both are relaxed, watching TV, I and my friend, one of us has sickle cell anemia, and both of us, we are relaxed, and we're just we're watching TV, you can never tell. But when both of us start running on the treadmill, when there is an oxygen demand, then the cells of the person that has sickle cell anemia, they will start to sickle when you need them the most. When you need them to start working, to carry more extra oxygen, that's when they start to sickle. Questions? Is that clear? Is this clear? Okay. There are multiple alleles, and that is with the blood groups. If you are blood type A or blood type B, sometimes blood type O does not have these antigens. There's incomplete dominance is when you have intermediate phenotypes, and these intermediate phenotypes you have like for example, a red plant with a white plant, and you don't get red or white. You get something like pink, something in the middle. Okay? Both could be co-dominant when there is at the surface of the red blood cell antigen A and B. They coexist. That's an example of co-dominance. Polygenetic or genetic inheritance, that's when genes are at two or more loci contribute to a single trait. So these are genes at two different loci, but they are responsible for one trait. Pleiotropy is when one gene produces multiple phenotypic effects. So that's in alkaptonuria, where there's a mutation of the gene on the chromosome 3. Okay. All right, we talked about that. So epigenetics, that's when gene expression is altered without changes in the base sequence. It could be passed to the offspring, and that's called epigenetic inheritance. So cancer, you need to understand when it comes to cancer, what's the difference between malignant and malignancy? What's the difference between invasion and metastasis? The most important concept to understand when it comes to cancer and malignancy is the difference between invasion and metastasis. What is the difference between invasion and metastasis? Invasion is when the cancer cell Obviously, both are cancerous. Invasion and metastasis are both hallmark features of a cancer cell. No benign cell is going to ever do metastasis or invasion. So now we all agreeing that this is a cancer cell. So what's the difference between invasion and metastasis? Invasion is when the cancer cell is infiltrating the basement membrane and is spreading locally. So you have a cancer cell in the bone tissue, God forbid. A cancer cell in the lung tissue, God forbid. Cancer cell in the brain tissue, God forbid. If it remains there, then that's invasion. If it travels through, you see it infiltrated, that's the blue cancer cell. It broke into the blood vessel and traveled very long distance from the bone to the kidney, from the lung to the bone, from the liver to the brain. So that's not invasion anymore. That's now metastasis. That is metastasis. Okay. So carcinogens are substances that cause cancer. I mentioned one earlier. What's a substance that causes cancer? Besides processed food and high sugar and all of this unhealthy food. What else? Stress. What else? I mentioned earlier something. The ultraviolet radiation, the sun, can damage the DNA. Yeah, the melanin. Yeah, very good. Okay, any questions? Any questions? So that's a Wilms tumor. That's an example of a kidney tumor. Any question?